Hi, my name is Tristan Pierce. I will be presenting on the paper Evoked Itch Perception is Associated with Changes in Functional Brain Connectivity by Desbordorzas et al. Now to cover some background. Not much is known about the circuitry involved in itch perception. There have been studies that suggest pain perception is associated with altered resting state functional brain connectivity, and even a study regarding itch perception for itch-induced healthy patients. However, until this study, it is believed that there hasn't been any research on itch perception in patients with atopic dermatitis or AD. So before conducting this research, the question was posed, how is functional brain connectivity, specifically in AD patients, modulated by clinically relevant itch? Ultimately, they're asking what the pathway through the brain is for itch sensation. Methods that researchers used in this project included an initial screening for participants that included an extensive criteria for participation. fMRI scans and data was used to measure and map brain activity before and after procedural sessions. A baseline was determined weeks before the patient participated in itch induction. In addition, a baseline resting scan was performed at the beginning of the induction as well as following the session to determine the allergen-invoked itch state. Data collected from the fMRI was used to form a seed-based connectivity analysis. I will explain that on the next slide. But also, in addition, a visual analog scale was performed following the itch induction procedure to test the individual's perception of itch. So the temperature modulated itch induction procedure aided in measuring the brain activation of itch sensation, simply explained as a prick allergen test that includes cooling and heating sensations. And this test aided in viewing the different brain regions that activated during increased itch, peak itch plateau, and the reduction of itch period. And during the increased itch phase, the right anterior insula, the right anterior middle cingulate cortex, the bilateral striatum, the right globus pallidus, and the right ventrolateral prefrontal cortex were active. Then, in the peak itch plateau phase, there were three regions that were active: the right dorsolateral prefrontal cortex, the bilateral premotor cortex, and the left superior parietal lobule. Information was also provided for the reduction of the itch in the putamen and right anterior insula. As spoken of before, seed-based functional connectivity analysis is gathered from the fMRI scans. The seed is a region of the brain that is activated during the itch sensation and perception. The connectivity analysis is then used to construct a three-dimensional computer-based model of the seed that is under investigation. The seed-based correlation methods evaluate the changes in brain connectivity. Data from several regions was acquired, but I will only share an example of rest and itch. So in the image on the left, you can see the hippocampal formation as well as the posterior cingulate cortex are regions of the brain that are at rest because the clusters in those regions are a blue color. Whereas in the image on the right, the dorsolateral prefrontal cortex, you can see the clusters are red, which means it's indication of itch. And we see the difference in connectivity from rest to itch as a function of the seed-based functional connectivity analysis. The research findings were that a decreased functional connectivity from baseline resting to the evoked itch state between several itch-related brain regions, including the insular cingulate cortices and the basal ganglia. Also, decreased connectivity correlated with increased levels of perceived itch. And invoked itch increased connectivity between key nodes of frontoparietal control network, and that the higher connectivity correlated with a lesser increase in perceived itch. So the take-home message from this paper is that neurocorrelates of allergen-induced itch in AD involve a broad network of brain regions involving emotional processing, salience, executive functioning, sensory motor integration, and motor planning. This is my sources, and thank you for watching.